Hello dear students, today we are going to learn about primitive streak. So, which is one of the most important event which occurs in third week of development. So, in the second week we have seen that this bilaminar germinal disc is formed where this is the epiblast cells and these are the hypoblast cells forming the bilaminar germinal disc with the amniotic cavity above and yolk sac below. And this is the connecting stalk which is attached to the caudal end of this bilaminar germinal disc. Now this bilaminar germinal disc is initially circular. Now this is the view where we are viewing the embryonic disc from above or from the dorsal aspect or from this amniotic cavity side. So this is the bilaminar germinal disc with the connecting stalk which is attached at the caudal end of this disc. Now the shape of the embryonic disc changes from circular to oval, and then from oval on the 18th day of embryonic life it becomes pear shaped. That means it is broad in its cranial part and narrower in its caudal part. Now on the 15th day of the embryonic life the primitive streak is formed. You can remember this where if you count the letters in the primitive streak they are 15. So primitive streak is formed on the 15th day of the embryonic life. Now what is primitive streak? So this is the linear opacity. Primitive streak is the linear opacity which is formed in the caudal region of the embryonic disc on the dorsal aspect. That means on the amniotic side cavity. Now how it is formed? So this above germ layer, as you know, this is the epiblast cells. So these epiblast cells in the caudal region of the embryonic disc, they are pluripotent in nature. They will start migrating and proliferating simultaneously, come into the center of the caudal region of embryonic disc. They heap up in this region and form a linear opacity which is called as a primitive streak. So this is our primitive streak. Now in the cranial end of the primitive streak a circular elevation is formed which is called as a primitive node, also called as Henson's node. So this is the primitive streak and this is the Henson's node or primitive node. Now later on what happens? A groove develops in the primitive streak which is called as the primitive groove, shown here in the pink color, and this groove also continues in the primitive node which is called as a primitive pit. So this pink color is a primitive groove and this is the primitive pit. This primitive pit is also called as blastopore in the lower vertebrates or in the lower animals. This primitive pit is also a very important structure as it gives rise to the formation of notochord. Now we will see how the third germ layer is let down by the primitive streak in the third week of embryonic life. So in the third week of embryonic life this bilaminar germinal disc changes into trilaminar germinal disc. Trilaminar germinal disc. The formation of the third germ layer and this process is called as gastrulation. This process is called as gastrulation and the embryo which now turns from bilaminar to trilaminar is called as a gastrula. So how this third germ layer is laid down? Now this is the primitive pit. We have taken a cut section over of the embryo in this region. So we can see that this above blue color is the epiblast cells, and this brown colored cells are the hypoblast cells. Now what happens when, so here in this region is a primitive groove, as you know. Now when these cells, epiblast cells, come to the edge of this groove, to the edge of the primitive groove, the intercellular junctions between these epiblast cells, they loosen up, the shape of the cells change, they become flask shaped, and they start ingressing or invaginating between the epiblast and the hypoblast. So they are ingressing or invaginating between the epiblast and hypoblast. Now first they will replace these hypoblast cells. So first they will replace these hypoblast cells, so which we call it as a primary endoderm. And when this epiblast cells ingress and replace this hypoblast cells totally, the germ layer which is formed is called as a secondary endoderm or definitive endoderm. So this is the first germ layer which is being formed. So which is the first germ layer which is formed? Is the endoderm. Then what happens? These epiblast cells, after the formation of the definitive endoderm, they will occupy the space between the epiblast and the hypoblast, and this leads to the formation of the third germ layer, which is the mesoderm. So this pink color is the mesoderm. So as it is present within the embryo, it is called as intraembryonic mesoderm. Within the embryo, intraembryonic, or also called as definitive or secondary mesoderm. Now after the formation of the secondary mesoderm or intraembryonic mesoderm, whatever cells are there which are remaining in the this epiblast, whatever epiblast cells which are remaining, they will form the definitive or the secondary ectoderm, and this is how all the three germ layers are being formed. So you can say that all the three germ layers are derived from which cells? That is the epiblast cells. First they will replace the hypoblast, will form the definitive endoderm, then they will occupy the space between epiblast and definitive endoderm and will form intraembryonic mesoderm, and whatever cells which are remaining, they will form the definitive ectoderm. So this is how all the three germ layers are derived from the epiblast of the primitive streak. Therefore it is a very, very important structure, and this is the most important event in the third week, where the trilaminar germinal disc is being formed. Now the primitive streak, what are the functions or importance of the primitive streak? So as we can see here, it acts as a primary organizer. Primary organizer. That means it leads to the, or it induces the formation of the intraembryonic mesoderm, and it also induces, this primitive pit induces the formation of notochord. So this is how it acts as a primary organizer. So it induces the formation of intraembryonic mesoderm, and it induces the formation of notochord. Okay. Then it also acts as an axis. It forms the axis of the embryo, which gives rigidity to the embryo, and it determines its right left side, dorsal ventral side and cranial caudal ends of the embryo. So it also forms as the axis of the embryo, and it gives rise to all the 3 germ layers. So this is the importance of the primitive streak. Now when it regresses, as we have seen, it is formed on the 15th day of the embryonic life, and after its function is over, it will regress at the end of the 4th week, or to be more precise, it will regress on the 26th day of embryonic life. Okay? And as it regresses, the notochord will increase, or it is formed in the caudal region. It forms in the caudal region more. So it regresses on the 26th day, and if it fails to regress at the end of the 4th week, it leads to the formation of a tumor which is called as sacrococcygeal teratoma. Sacrococcygeal teratoma. As the name is suggesting you that it is present in the sacrococcygeal region of the baby which is born. And why teratoma? We have seen that it is formed by the pluripotent epiplast cell. So it has got all types of the tissue, and as it is present in the sacrococcygeal region, it leads to obstructed labor, and it is more common in female babies. The reason is not known, and it has to be removed within 6 months after birth. So that's all about the primitive streak. To watch such more videos, educational, exam oriented educational videos, watch my channel or subscribe my channel.